Hello friends, welcome to the second part of lecture on proteins. Here is another question on the proteins. The exact backbone dihedral angles in a folded protein can be obtained by, the options are given, deconvolution of its circular dichroism spectra obtained at different pH and temperature, estimating the number of protons that exchange with deuterium on treating the protein with D2O, forming fibers of the protein and analyzing the fiber diffraction pattern, analysis of the crystal structure of the protein obtained by X-ray diffraction at high resolutions. Another question was also asked in June 2019. This was related to the protein folding. The question is, thermodynamics of protein folding is depicted as a free energy funnel below, and the image is shown where the entropy level is given. It is shown to increase from down to up, and the percentage of the protein in native conformation is shown. This is the 0% of protein in native conformation, and this is the 100% protein in native conformation. And on the basis of this figure, certain uh, uh, two columns are given where A, B, C, D are shown over here in the X column. And what does this A indicate? Whether it is indicating the native structure or structure with highest entropy or the molten globule, discrete folding intermediates, all these are shown. So we have to correlate this percentage of the protein in native conformation with the entropy. So let us move for the discussion on the remaining aspects of the protein first, and then we'll come to the discussion on these questions. Simple and conjugated proteins. Simple proteins are those consisting of only amino acids such as serum albumin. Conjugated proteins, they consist of simple proteins which are conjugated with non-protein component such as glycoproteins. Examples include fibronectin, cadherin. Lipoproteins are the proteins in which the lipids are conjugated with the proteins. The examples include chylomicron, high density lipoproteins, and so on. Metalloproteins includes the proteins which are conjugated with the metal ions. So ferritin is an example, alcohol dehydrogenase which has zinc in its structure, cytochrome oxidase which has copper and iron in its structure, nitrogenase complex which has molybdenum and ferrous in its structure. Hemoprotein is another class of the conjugated proteins where the proteins are conjugated with the heme group. The examples include hemoglobin, myoglobin, cytochrome C, catalase, etc. Proteins can also be classified into two major groups such as fibrous proteins and globular proteins. Fibrous proteins are those which have polypeptide chains arranged in long strands or sheets, and globular proteins having polypeptide chains folded into a spherical or globular shape. The two groups are structurally distinct. Fibrous proteins usually consist largely of a single type of secondary structure. Globular proteins often contain several types of secondary structure. The two groups differ functionally in that the structures that provide support, shape, and external protection to vertebrates are made up of fibrous proteins, whereas most enzymes and regulatory proteins are globular proteins. Here is the table indicating the secondary structures and properties of the fibrous proteins. You can see that in some of the fibrous proteins, only single type of secondary structures are found. For example, alpha helix cross-linked by disulfide bonds. These are found in the keratin of hair, feathers, and nails. And uh, these structures, they provide tough, insoluble protective structures of varying hardness and flexibility. This is the characteristic of such proteins. Then the beta conformation, only beta conformation is found in proteins like silk fibroin, and the characteristic of this silk fibroin is that it is soft, flexible filaments. Collagen triple helix is another kind of secondary structure which is found in collagen of tendons, bone matrix, and the characteristic of this protein is that they have high tensile strength without stretch. Small globular proteins include myoglobin, then hemoglobin. So let us discuss first about myoglobin. Myoglobin is a relatively small, molecular weight is 16,700 daltons. It is an oxygen binding protein of muscle cells. It functions both to store oxygen and to facilitate oxygen diffusion in rapidly contracting muscle tissue. Myoglobin contains a single polypeptide chain of 153 amino acid residues of known sequence and a single iron protoporphyrin or heme group. The same heme group is found in hemoglobin, the oxygen binding protein, which is found in erythrocytes and is responsible for the deep red-brown color of both myoglobin and hemoglobin. Myoglobin is particularly abundant in the muscles of diving mammals such as the whale, seal, and porpoise, whose muscles are so rich in this protein that they are brown. Storage and distribution of oxygen by muscle myoglobin permit these animals to remain submerged for long periods of time. Here are some examples of other proteins, and these globular proteins, they have both the kind of structures, the uh, alpha structures as well as beta conformations. They are also found. Both the type of structures, secondary structures, are found in the globular proteins. So you can see that in the chymotrypsin, the 14% of the amino acid residues are in alpha helix conformation, whereas the 45% of amino acid residues are in beta conformation. Ribonuclease is another globular protein which has 26% of amino acid residues in alpha helix conformation and 35% amino acid residues are in beta conformation. Myoglobin, it has 78% in alpha helix conformation and none are found in the beta conformations. Hemoglobin is another globular protein whose molecular weight is 65,450 daltons. It is an oligomeric allosteric conjugated protein with 4 polypeptide chains joined by non-covalent bonds. It contains heme prosthetic group, which is an iron-containing porphyrin derivative. Hemoglobin is present in all vertebrates except Arctic fish and in many vertebrates including annelids, many arthropods, and some mollusks. Human hemoglobin, called as HbA1, it consists of 4 polypeptide chains of two types, two alpha chains and two beta chains, and hence it is often called as alpha to beta 2 hemoglobin. The polypeptide portion is collectively called as globin. Alpha chains contain 141 amino acid residues and beta chains contain 146 amino acid residues. Other forms of hemoglobin are also found, for example, embryonic hemoglobin and fetal hemoglobin. Embryonic hemoglobin contains two sigma chains which are similar to alpha chains and two epsilon chains which are similar to beta chains. Fetal hemoglobin contains two alpha chains and two gamma chains which are similar to beta chains. Both embryonic and fetal forms have a higher affinity for oxygen, which is necessary to obtain oxygen from the mother's blood. Hemoglobin binds to the oxygen in a sigmoidal manner because it binds to the oxygen in a cooperative manner. Now this figure indicates that the partial pressure of oxygen in the lungs is near about 13.3 kilopascals, whereas the partial pressure of oxygen in the tissues is around 4 kilopascals. So if the hemoglobin is in low affinity state, it won't be able to bind to the oxygen in the lungs. And if it is in very high affinity state, such that the myoglobin is very uh, high or tightly binding protein to the oxygen, it has more affinity towards oxygen compared to hemoglobin. So myoglobin won't be able to serve this function of transporting the oxygen from lungs to the tissues because its affinity towards oxygen is very high, and at a low level of oxygen also the oxygen will remain tightly bound to the myoglobin, whereas hemoglobin can serve this purpose of transporting oxygen from lungs and delivering the oxygen to the tissues, because when it transits from low to high affinity state, still it can bind to the oxygen in the lungs and it can then deliver or it can release the oxygen in the tissues. So hemoglobin is a better oxygen transporter protein molecule compared to myoglobin, which is found in the muscles. Protein folding. Protein folding is the physical process by which a polypeptide folds into its characteristic and functional three-dimensional conformation. The correct three-dimensional structure is essential to protein function. Failure to fold into native structure produces inactive proteins. A protein molecule folds spontaneously during or after biosynthesis. However, the process also depends on the nature of solvent, the concentration of salts, temperature, and the presence of molecular chaperones. Molecular chaperones. Molecular chaperones are a class of proteins which bind to incompletely folded or unfolded proteins in order to assist their folding or prevent them from aggregating. Chaperones prevent the formation of incorrect structures rather than promoting the formation of correct structures. Chaperones may also be required to assist the refolding of stressed denatured proteins, formation of oligomeric structures, protein trafficking through membranes, and assistance in proteolytic degradation. Examples of molecular chaperones include HSP protein, which is called as heat shock proteins, as they are synthesized in increased amounts after a brief exposure to elevated temperature. These were discovered in E. coli and they are also found in the eukaryotic cells. Chaperones are classified according to their molecular weight. For example, HSP40 has 40,000 Dalton, HSP60 has 60,000 Dalton, and so on. Two families of HSP are found, HSP60 and HSP70. Members of these two families, they function differently. HSP70, these function during protein synthesis. Members of HSP70 family, such as HSP70 itself, HSC70, HSP40, GRPE, they act they act early in the life of many proteins, binding to a string of about 7 hydrophobic amino acids before the protein leaves the ribosome. The HSP70 polypeptide chain is divided into two functional regions, one that binds and hydrolyzes ATP, and a second that binds hydrophobic segments of unfolded polypeptide chain. The polypeptide binding domain is an antiparallel C-terminal region. HSP70 is induced by stress, example heat shock, whereas HSC70 is constitutively expressed in the cells. Cytosolic HSP70s, they prevent misfolding and maintain the polypeptide chain in unfolded condition. They are also needed for the normal translocation of protein from cytosol into either endoplasmic reticulum or mitochondria. HSP70, it is also called as DNA-K in E. coli. It works in tandem with HSP40, which is also called as DNA-J protein in E. coli. The ATP-dependent reaction cycle of HSP70 is regulated by the HSP40. Hydrolysis of ATP to ADP is strongly accelerated by HSP40. Here is the figure indicating how HSP70 family members they function. The protein is shown after synthesis. The protein which is being synthesized, it generally folds and the folding intermediate is formed before it folds into the native conformation, which is a functional one. It may happen that if the during folding the incorrect folding occurs, then these incorrectly folded protein structures they accumulate and they are seen as aggregates. Now this HSP70 family, these remain attached to the ATP, and when bound to ATP, they have low affinity towards the protein. But HSP40, when it joins with this HSP70, it catalyzes the hydrolysis of ATP to ADP, resulting in the conformational change in the HSP70 from low affinity to high affinity state, and the protein gets tightly bound to the HSP70, and this causes the partial folding of the protein aided by the HSP70 molecular chaperones. And this partially folded protein is then released, and which generally folds into a native conformation after it has been partially folded by HSP70. And later on, the nucleotide exchange factor causes the exchange of ADP with ATP, resulting in con, uh, again conversion of the HSP70 ADP into HSP70 ATP complex, which is again available to bind another molecule of polypeptide chain for its folding into partially folded structure. So this is how HSP70 along with HSP40, they cause the proteins to fold properly. If the folding is not proper, then again the folding intermediate can be again bound by the HSP70 family and the folding can be carried out. And yet if it is not possible, then these HSP70 ADP molecules, they deliver the protein to HSP60 family, which then fold the proteins in a proper manner. HSP60 family of molecular chaperones, these are also called as chaperonins. HSP60 family form a large barrel-shaped structure that acts later in a protein's life after it has been fully synthesized. Chaperonins bind unfolded, partly folded, and incorrectly folded protein molecules, but not protein in the native state. Examples include HSP60, which is also called as GRO-EL in E. coli, consists of 14 subunits arranged in two heptameric rings stacked on top of each other in an inverted orientation. HSP60 associates with a ring-shaped heptamer of HSP10, which is called as GRO-ES in E. coli, which is also called as co-chaperonin. So here the partially folded protein is delivered by HSP70 ADP complex to the GRO-EL protein, which is a barrel-shaped structure, and two units are shown to be associated with each other, and the GRO-ES then combines with the GRO-EL, which is also called as HSP60, and GRO-ES is called as HSP10. So after the protein is delivered into this barrel, then GRO-ES binds to the GRO-EL protein like a cap, and the ATP hydrolysis occurs. During this, the proper folding of the protein occurs, and the protein is then released. After the protein has been properly folded, then the protein is released. On the other hand, the another HSP70 ADP complex can deliver the protein to the other side of the HSP60, and this can be followed by the binding of ATP molecules, and the ATP hydrolysis occurs to ADP and so on. So during this, the hydrolysis of ATP will cause the chaperonins to cause the proper folding of the protein into the native form, and this native form is then allowed to or is made available to do its function. So this is how the chaperonins, they cause the proteins to fold properly. Chaperonins are of two types: group 1 chaperonins, which includes HSP60s in mitochondria and chloroplast of eukaryotes, and GRO-EL in eubacteria. These have 7-membered rings and functionally cooperates with HSP10, which is also called as GRO-ES in eubacteria. Group 2 chaperonins, these are found in archaea and eukaryotes cytosol. They have 8-membered rings, and these are independent of HSP10 proteins. Protein sequencing. Determination of amino acid compositions can be done, or N-terminal amino acid analysis can be done. Admin degradation, which is responsible for the sequencing of protein, and the proteins may be subjected to cleavage with the uh, proteolytic enzymes, and then the amino acid sequence can be deduced from the peptides which are formed. So let's discuss about these protein sequencing methodologies. Determination of amino acid compositions. For this, the peptides are hydrolyzed with strong acid or base. Acid hydrolysis is carried out with 6 molar HCl at 110 degrees Celsius for 24 hours, whereas base hydrolysis of the proteins is carried out by heating with 2 to 4 molar NaOH at 100 degrees Celsius for 4 to 8 hours. Amino acids in the hydrolyzed states are then separated by ion exchange chromatography or by reversed phase HPLC. Amino acids are then identified by its elution volume and quantified by reaction with Nene-Hydrine. N-terminal amino acids analysis can be done with the help of reagents such as FDNB, densyl chloride, dapsyl chloride. These reagents they bind to the amino terminal or free amino group of the N-terminal amino acid. So here 1-fluoro-2,4-dinitrobenzene called as FDNB. It is also called as Sanger's reagent. It is when the peptide is allowed to react with FDNB. FDNB binds to the amino group of the amino terminal amino acid, resulting in the 2,4-dinitrophenyl derivative of the amino terminal amino acid residue. This, upon treatment with the mild uh, acid, it can cause the release of this amino terminal amino acid, and the other uh, the other amino acids in the peptides also can be broken down. But this amino terminal amino acid residue will be present in the dinitrophenyl derivative form, and this can be then identified through electrophoresis or chromatography techniques. Edmond degradation is used to carry out the sequencing of protein, where the protein is treated with phenyl isothiocyanate at high pH. So this will cause the binding of the phenyl isothiocyanate to form phenyl thiocarbamyl derivative, and this uh, this phenyl isothiocyanate will bind to the amino terminal of the amino acid and terminal amino acid residue in the peptide chain, and this is then followed by the treatment with the anhydrous trifluoroacetic acid at and acidic pH. This will cause the release of this thiazolinone derivative of N-terminal amino acid residue, and the rest of the peptide will be there, which is shortened by one amino acid. And slowly at low pH, this is converted into phenyl dihydrogenone derivative of the amino acid residues, which can be then extracted with the solvent and is then identified uh, through the appropriate technique. And again, the remaining peptide is purified, and the recycling of the remaining peptide fragment through Edmond process is again carried out. And this is how sequential removal of the amino acid is done, and the thio phenyl thiohydrantoin derivative of the respective amino acids are then identified through electrophoresis or through chromatographic techniques. The peptides can be subjected to cleavage with the proteolytic enzymes. These include trypsin, chymotrypsin, pepsin, cyanogen bromide. Cyanogen bromide is not a proteolytic enzyme, it is a chemical, whereas others are the enzymes listed in the table. Trypsin, now these enzymes are highly specific in their action. They cleave the peptide bonds at a particular amino acid residues. For example, trypsin, it cleaves at the carboxyl side of the lysine and arginine, whereas chymotrypsin, it cleaves at the carboxyl side of phenylalanine or the carboxyl side of tryptophan or carboxyl side of tyrosine. Pepsin, it causes the peptide bond cleavage on the amino side of the leucine or phenylalanine or tryptophan or tyrosine. Cyanogen bromide causes the peptide bond to be hydrolyzed at the carboxyl side of the methionine. So this property of these proteolytic enzymes or the cyanogen bromide can be used to cleave the polypeptide chain or the peptide chain into small peptides, and then the sequence of the amino acid can be deduced. Here is an example of a small peptide, and the peptide sequence is given, and if you are asked to indicate the number of bonds that could be hydrolyzed by digestion with trypsin or chymotrypsin or cyanogen bromide, then you must be able to identify that on which bond the trypsin will act, or on which peptide bond the chymotrypsin will act. So let's discuss. Trypsin, it acts on the carboxyl side of lysine or arginine. So there is no lysine or arginine, and so no peptide bond can be hydrolyzed by trypsin in this case. Another case is chymotrypsin. Chymotrypsin, it acts on the carboxyl side of phenylalanine, tyrosine or tryptophan, and so you can see there is one tyrosine and one phenylalanine. So carboxyl side of the phenylalanine will be cleaved and tyrosine also will be cleaved. So these two peptide bonds can be cleaved by chymotrypsin. Cyanogen bromide, it causes the peptide bond to be cleaved at the carboxyl side of the methionine. Now on the carboxyl side, there is no other amino acid linked to methionine, and so there is no peptide bond. So no bond can be broken down by cyanogen bromide, because cyanogen bromide cleaves after methionine, and that after methionine there is no amino acid. So this was about the peptide sequencing with the help of proteolytic enzymes. Purification of proteins. The basic aim of purification of protein is to isolate the particular protein of interest from the mixture of proteins. Different features such as solubilities, size, charge, adsorption properties are utilized during purification process. Here are the certain methods which are based on certain properties of the proteins that are shown over here. For example, centrifugation, gel filtration, dialysis and ultrafiltration, gel electrophoresis, these all make use of the size or mass of the proteins. Whereas ion exchange chromatography, electrophoresis, isoelectric focusing, they make use of charge of the proteins. Whereas the change in pH, change in ionic strength, decrease in the dielectric constant for causing the precipitation of the protein, that makes use of the solubility characteristic of the proteins. Whereas affinity chromatography makes use of the specific binding of the protein uh, to the particular resins. So here you can see that several separation methods can be used for purification of the proteins, and as the proteins get purified, the activity of the protein can be increased in the sample. So if it is an enzyme, then the specific activity of the protein sample will increase as the purification increases. So here in case of enzymes, the specific activity is monitored, whereas if the protein is not an enzymatic is not an enzyme, then other properties of the protein can be used for monitoring the purification level of the proteins during the purification steps. So let us discuss about the question. The exact backbone dihedral angles in a folded protein can be obtained by deconvolution of its circular dichroism spectra obtained at different pH and temperature, estimating the number of protons and exchange with deuterium on treating the protein with B2O, forming fibers of the protein and analyzing the fiber diffraction pattern, analysis of the crystal structure of the protein obtained by X-ray diffraction at high resolutions. So here circular dichroism can be used for predicting the secondary structures of the protein, whereas the forming fibers of the protein and analyzing the fiber diffraction pattern This cannot be obtained in this case. And for obtaining the exact dihedral angles, the X-ray diffraction is most appropriate one. So analysis of the crystal structure of the protein obtained by X-ray diffraction at high resolution is the most appropriate option for this question. In this question, the thermodynamics of protein folding is depicted as a free energy funnel below. So here the figure is shown and the different forms of the protein are shown. That is the protein which is in native structure, or it is in molten globule form, or discrete folding intermediates are there. So which alphabet is indicating which structure or which conformation of the protein that we have to predict. On this scale, the percentage of the native protein in the native conformation is shown, and we have to now predict the which conformation is for the native structure and so on. So on this in this column X, the A, B, C, D, these are shown, and on the next column the different forms of the different conformations of the protein are shown. The native structure with highest entropy, then molten globule, and discrete folding intermediates. So here you can see that this is the uh, protein which is in totally unfolded form. Okay, that is 0% of the protein is in native conformation. That means the entropy or level of this conformation will be highest one. Okay, so A is the structure with highest entropy, and D is the one which is 100% in the native conformation. So the native structure D1. So A2 D1 is there. Okay, so A2 and D1 is in the first option. And let us look at the other also. B is the one which has the intermediate one. Okay, so molten globule can be regarded in this case, and C1, C is the discrete folding intermediates. So C4 is the most appropriate. So the most appropriate option is the first one, A2, B3, C4, and D1. So this is the correct option. So this was about the proteins, and uh, the this is my references which I have used in this PPT. And so this was all about proteins. Thank you and happy learning.